Welcome to G Fun Facts Online. In the grand story of human evolution, few moments are as pivotal as when our ancestors first stood up and walked on two legs. This wasn't just a change in posture, it was a revolutionary leap that set our species on an entirely new path. Bipedalism freed our hands to create tools, carry our young, and gesture, paving the way for technology, complex societies, and even language. For centuries, we pieced this story together using the silent testimony of fossilized bones. But now, a new field of science is allowing us to read a much older and more intimate script, the one written in our own DNA. Today, we're diving into the world of paleogenetics to uncover the precise genetic mutations that re-engineered our bodies for a two-legged world. Before we get to the genetic code, it's important to appreciate the sheer scale of the anatomical revolution that bipedalism demanded. Our quadrupedal primate ancestors had a skeleton perfectly adapted for life in the trees. Transforming this into a body that could balance and stride efficiently on the ground required a head-to-toe overhaul. The fossil record gives us a timeline of this transformation. Between 4 and 7 million years ago, the first tentative signs of this shift appear. The discovery of a species called Sahelanthropus chidensis, which lived around 7 million years ago, has sparked debate. Some scientists argue that the position of its foramen magnum, that's the hole at the base of the skull where the spinal cord connects, suggests a more upright posture than seen in other apes. By the time of the Australopithecines, a group that includes the famous Lucy skeleton, the evidence for bipedalism is much clearer. These early hominins, living between 2 and 4 million years ago, had a whole suite of adaptations for upright walking, even if they retained some features for climbing. Let's break down these key anatomical changes. Perhaps the most dramatic change happened in the pelvis. It transformed from the tall, narrow structure seen in our chimpanzee cousins into a short, wide, bowl-shaped basin. This new shape provided stability for upright walking and running. It did this by repositioning the gluteal muscles to the side of the hip joint. This allows them to steady the pelvis as we shift our weight from one leg to the other, a crucial part of the bipedal gait. Next, the spine. To support an upright torso, the human spine developed a series of curves, an S shape, that acts like a spring. It absorbs the shock of walking and keeps the head and trunk balanced perfectly over the pelvis. Our legs also became longer in proportion to our arms, which increased our stride length and efficiency. The femur, or thigh bone, angles inward from the hip to the knee. This brings our feet closer to the body's midline and further enhances our balance. Then there's the foot. It was once a grasping structure, almost like a hand, but it was reshaped into a rigid platform for pushing off the ground. The big toe became aligned with the other toes, losing its ability to grasp, and a prominent arch developed to absorb shock and provide a spring in our step. Finally, as mentioned with Sahelanthropus, the foramen magnum migrated to a more central position at the base of the skull, allowing the head to balance directly on top of the spinal column. These weren't minor tweaks. Each change was a profound evolutionary commitment, a step away from the trees and toward a new life on the ground. For a long time, how these changes occurred was a mystery. But now, paleogenetics is starting to identify the genetic switches that were flipped along the way. Paleogenetics is a field that would have been science fiction just a few decades ago. It involves recovering and analyzing DNA from ancient remains. Now, getting usable DNA from fossils that are millions of years old, the time frame when bipedalism first emerged, is still largely impossible. But scientists have developed some ingenious workarounds. The primary tool for studying deep evolutionary history is comparative genomics. By comparing the genomes of modern humans with those of our closest living relatives, chimpanzees, bonobos, and gorillas, researchers can identify sections of our DNA that are uniquely human. These are the regions that have changed significantly since our lineage split from that of other apes, and they are prime hunting grounds for the genes that make us human, including those for bipedalism. Another powerful technique is the genome-wide association study, or GWAS. In a GWAS, scientists scan the complete genomes of thousands of individuals, looking for tiny variations in the DNA sequence, known as SNPs. They then see if any of these SNPs are more common in people with a particular trait. By applying this to skeletal characteristics, researchers can pinpoint genes that influence the shape and proportions of our bones. One of the most exciting discoveries from this field is the identification of human accelerated regions, or HARs. These are short stretches of DNA that are highly conserved across most mammals, meaning they've changed very little over millions of years. But in the human lineage, these regions have undergone rapid and dramatic changes. This strongly suggests they're associated with uniquely human traits, like brain development and, you guessed it, bipedalism. Now let's get back to the pelvis, the keystone of our upright anatomy. A landmark study, hypothetically published in 2025, identified two crucial innovations in how the human pelvis develops and pointed to the genetic machinery behind them. The first innovation was a dramatic 90-degree rotation of the ilium's growth plate during embryonic development. The ilium is that large, wing-like bone you can feel at your hips. In other primates, the ilium grows to be tall and flat. But in human embryos, the growth plate, where new bone is formed, radically shifts its orientation. This causes the ilium to grow sideways, creating the characteristic short, wide, and curved bone that is so essential for our balance. This shift isn't the result of a single bipedalism gene. Instead, it seems to be orchestrated by a network of regulatory genes, DNA switches that control the activity of other genes. Researchers identified several key players, including a gene called SOX9. This is a master regulator of cartilage formation. Mutations in SOX9 can cause a rare genetic disorder that results in abnormally narrow hip bones, lacking the human flare. This provides a direct link between this gene's function and the bipedal shape of the pelvis. Another gene, PTH1R, is also critical, as mutations in it also lead to abnormally narrow hip bones. This first big change, the sideways growth of the ilium, is thought to have occurred early in our lineage, around the time we split from the ancestors of chimpanzees, between 6 and 8 million years ago. It was a foundational change that set the stage for everything that followed. The second major innovation was a delay in the ossification of the pelvis. Ossification is the process where soft cartilage hardens into bone. Researchers discovered that in humans, the ilium is significantly slower to turn into bone compared to the rest of the skeleton, lagging by about 15 weeks. This delay is thought to have evolved much more recently, perhaps within the last 2 million years, and it likely came about as a solution to a classic evolutionary problem known as the obstetrical dilemma. The dilemma is this: efficient bipedalism favors a narrow pelvis, but the evolution of our large brains requires a wide birth canal for childbirth. By delaying the hardening of the pelvis, our ancestors found a clever way around this. The extended period of cartilage growth allowed the pelvis to continue to expand, preserving the wide, bowl-like shape needed for walking, while also accommodating the birth of big-brained babies. Key genes implicated in this delayed timing include RUNX2 and FOXP1/2, which act as conductors for this specific ossification pattern. Together, these two shifts, the rotation of the growth plate and the delay in ossification, represent a complete overhaul of how the pelvis is built. They show a core principle of evolution. Profound anatomical changes often arise not from inventing entirely new genes, but from subtle tweaks to the timing and location of the activity of existing ones. But the story doesn't end at the pelvis. A 2023 study published in Science used deep learning algorithms to analyze over 30,000 full-body X-rays. By training the algorithm to precisely measure a wide range of skeletal features, researchers were able to conduct a massive GWAS to find the genetic variants that influence our skeletal proportions. This research identified 145 key points in the genome that control the dimensions of our skeleton, from the width of our shoulders to the length of our legs. This provides, for the first time, a genomic map of the skeletal changes that enabled the transition from knuckle walking to bipedalism. The study found that many of these genomic regions show signs of having been under strong natural selection, providing the first direct genomic evidence that there was evolutionary pressure on these genetic variants. The findings also revealed that many of these genes are located in those human-accelerated regions we talked about earlier, further cementing their role in our unique evolutionary journey. Another fascinating area of research comes from studying rare genetic conditions. For example, a 2008 study of several families in Turkey, some of whose members walk on all fours, identified mutations in a gene called VLDLR. This gene is critical for the proper development of the cerebellum, a part of the brain crucial for motor control. While this condition is complex, it provides a window into how a single gene mutation can have a profound impact on gait and locomotion. But evolution is not a perfect process. It's a series of compromises and trade-offs. While walking upright brought enormous advantages, it also came at a cost. The same skeletal architecture that lets us stride across the globe also makes us uniquely susceptible to a range of musculoskeletal problems. The same deep learning study that identified the 145 genomic loci for skeletal proportions also made a startling discovery. Many of these same genetic variants are linked to an increased risk of arthritis. This suggests that the evolutionary changes that gave us bipedalism also left us with a legacy of aching joints. The correlations were specific. People with a higher ratio of hip width to height, a key feature of our bipedal pelvis, are more likely to develop osteoarthritis and pain in their hips. Individuals with a higher ratio of thigh bone length to height are more prone to arthritis in their knees. And those with a higher ratio of torso length to height are more likely to suffer from back pain. This makes intuitive sense. The biomechanical stresses on our joints are immense, and our skeletal proportions directly impact how those stresses are distributed over a lifetime. It appears that the very traits selected to make us efficient bipeds inadvertently created predispositions to these common and debilitating conditions. Our upright stance puts enormous pressure on our lower back, hips, and knees. The ultimate dream of paleogenetics would be to extract and sequence the entire genome of an early hominin like Lucy. This would give us a direct look at the genes active during the dawn of bipedalism. Unfortunately, DNA is fragile and rarely survives for millions of years. The oldest hominin DNA sequence to date is from a 430,000-year-old individual. The genetic record from 6 million years ago has long since turned to dust. But all is not lost. By studying the ghosts of these ancient genes in the genomes of living people and our primate cousins, we can reconstruct a surprisingly detailed picture. Scientists can identify genes present in humans but not in other apes, or genes with different patterns of regulation, and infer that these changes arose after our evolutionary paths diverged. This is exactly how researchers were able to pinpoint the developmental shifts in the pelvis. By comparing the process in human, chimpanzee, and mouse embryonic tissues, they could deduce the changes that must have occurred in our lineage. The story of how we came to walk upright is a testament to the power of evolution to reshape and reinvent. It wasn't a single event, but a long, complex process of anatomical and genetic tinkering over millions of years. For the majority of human history, it was our upright posture, not our large brains, that defined us. Thanks to the revolutionary field of paleogenetics, we can now read the story of our evolution in the living library of our own genomes. We are beginning to identify the very genes and regulatory switches that sculpted our pelvis, lengthened our legs, and set us on 2 feet. This new knowledge does more than just satisfy our curiosity about our origins. It provides a deeper understanding of our own bodies, our strengths, and our vulnerabilities. It explains why a mode of locomotion that allowed us to conquer the planet also leaves many of us with aching backs and knees. The upright switch was the first and perhaps most fundamental step on the journey to becoming human. And as the tools of paleogenetics become ever more powerful, we can be sure that many more chapters of this incredible story are still waiting to be read in the ancient and elegant code of our DNA. Thanks for joining us on Fun Facts Online.